Let's continue part 2 of the chapter 10, that is microbes in human welfare. So today we will discuss with the heading 10.2.3, that is some chemical enzymes and bioactive molecules that are secreted by these microbes. So microbes are used to produce chemicals for industrial processes. So basically for all the industrial processes we use these microbes for the production of various elements. Now we will have a table over here where we will first see the agent, means the one which is producing some action, then we will see its source or what is its type, and then we will see action, that what does it do. Aspergillus niger, which is a fungus, gives citric acid. Acetobacter aceti, which is a bacteria, gives acetic acid. Clostridium butylicum, which is a bacteria, gives butyric acid. Lactobacillus, which is again a bacteria, gives lactic acid. Saccharomyces cerevisiae, which is a yeast, produces ethanol. We have used this yeast earlier in the fermentation of various beverages. Streptokinase is derived from Streptococcus, which is used as a clot buster in human bodies. Cyclosporin A, which is derived from Trichoderma polysporum, is actually immunosuppressive reagent. Statins, which is derived from Monascus purpureus, decreases the cholesterol of the blood. Next heading that comes is the microbes in sewage treatments, and this is also very important heading. So here we are going to discuss about how the purification of sewage water is done with the help of microbes. So the sewage water, which consists of human excreta, is pathogenic, means it causes diseases. Therefore we need to treat it at sewage treatment plants. Short form is STP, and it is done by heterotrophic bacteria which are present already. Now this process is done in two steps: the primary treatment and then the secondary treatment. Primary treatment involves the physical removal of the solid via sedimentation and filtration. So this is the major aim of primary treatment. It removes all the solid particles, major of the solid particles, through the two processes, sedimentation and filtration. So in this, floating debris is generally removed by filtration and grit and pebbles are removed by sedimentation. Now after the removal of these two, the one now which has been settled is primary sludge, and the one which is flowing yet is primary effluent. This primary sludge is again removed, and this primary effluent we have to treat it further, as it will only give us water, pure water. This primary effluent is taken to a secondary treatment plant which has aerobic conditions. Aerobic conditions means in the presence of air and oxygen. So here in the secondary treatment plant, this primary effluent is passed into aeration tanks and the air is pumped into it. They are agitated in the presence of air. Due to this, it leads to the formation of flocks. Now due to this agitation of the air, the flocks formation take place. Now what is a flock? So flock are the masses of bacteria that are associated with fungal filaments, means it is a combination of bacteria and fungi to form mesh like structures. Now these flocks consume organic matter from that effluent. Now when it will consume all the organic matter, means the microbes which were earlier taking oxygen from that effluent are not present because they are eaten by flock. Now it will reduce the biochemical oxygen demand, that is BOD. This BOD indicates the polluting potential of any of the effluent. More the BOD, this means that more the oxygen demand in the effluent, more it will be polluting. So as these flocks decompose organic matter, the BOD eventually decreases and hence the polluting potential. Afterwards, the secondary effluent that is so formed is sent to settling tank. Now we can see this as a third or tertiary treatment, but yet we are considering just the name, that is settling tank. Here the flocks settle and form the inoculum. These flocks means these flocks will be taken in a few amount and will be again added to the secondary treatment, which will similarly repeat the process like we discussed of curd. Anaerobic sludge. Now this settling tank is also called as anaerobic sludge digester tank. After this settling tank, whatever effluent is left that from which flocks is removed is now sent to anaerobic sludge digester tank, wherever anaerobic conditions are taking place and anaerobically the other microorganisms are digested. Here bacteria and fungi are digested, means all the remaining organisms. Now where on the digestion these bacteria produce methane, hydrogen sulfide and carbon dioxide. These three combination gives us biogas. So afterwards we see that in anaerobic sludge digester tank we get biogas due to the digestion of bacteria. This is released in the water bodies now. The effluent so formed is very pure and every microorganism has been decomposed. Now there was a Ganga action plan and Yamuna action plan that was proposed initially to build a large number of sewage treatment plants in the various parts of country where there are the polluting water bodies, and then discharge the treated water only. So more will be the sewage treatment plants, more pure water will be formed and will be discharged, and there will be a less chance of degradation of water resources.